Hey guys, welcome back. So in this part of the video, this is the third part of the video on the liver histology. So in this part of the video, we're going to talk about the details of the liver lobular structure. So as I have mentioned before in the previous video, the liver parenchyma and its arrangement of hepatocyte can be explained in three ways. The first way is the classic lobule, the second way is the portal lobule, and the third way is hepatic acinus. So in this slide, we're going to talk about the classic lobule first. So before I move on, I'd like to explain about this diagram first. So I've mentioned before in the previous slide, so the blue represents the portal vein, and this one is a hepatic artery, and this one is actually the bile duct, and this is actually the lymphatic vessel. Okay. And these bars are actually showing that the sinusoids are actually leaky, and sorry, this okay, this is actually the endothelial cells of the sinusoid, the blue color one. I think I should use the blue color one is the endothelial cells of the sinusoids, and this one, the black color one, which is in dotted, they are the basal lamina of the sinusoids. As you can see, they are actually quite leaky. That's why they actually can move on to the content of the plasma can actually move on to the space of DC. And so whereas the one that is actually in the black color, this is actually the Kupffer cells, the macrophage that is inside the hepatocytes, and this is, as you know, the macrophage that is inside the liver, and this is the, as you know, this is the hepatocytes arranged in pods or plates, and hepatocytes are basically they are actually a large cell. They are actually 20 to 30 microns, and they are actually polyhedral cells with multiple faces, where one of the faces is actually facing the blood, another face is actually facing the canalicular system. And so how exactly is the structure of the sinusoids? Is the sinusoids are actually made up of discontinuous endothelial cells with fenestration. So this is actually the endothelial cells, the blue color with fenestration, the in between one. And the basal lamina is also discontinuous, as you can see, the black color one, the dotted one. So within sinusoids, we have Kupffer cells. Okay, and these Kupffer cells are derived from the circulatory monocytes or macrophages. And they line the endothelial cells so that when blood moves from the periphery to center, the components of the blood are actually freely exposed to the Kupffer cells. So Kupffer cells, that's why the Kupffer cell actually can remove the remnants of red blood cell from the blood blood carried from the spleen. At the same time, they also can filter the bacteria. So now we're going to move into hepatocytes. Okay, we will look into hepatocytes in greater detail. The next slide. Okay, so I've labeled everything enough here, so I'm not gonna explain these figures, but I'll move on to talk about. Okay, this slide is actually very important. Try to pay close attention to this. So okay, as you can see here, this is actually the space of DC. So this side is exposed to compounds, components of the plasma, and the face of the hepatocytes is also facing towards the space of DC, as you can see here. Okay, and as for the hepatocytes, they have multiple villi. Okay, this is actually the villi to increase the surface area. Okay, then bile canaliculi. Okay, so basically bile canaliculi are basically just gaps between the hepatocytes in one pod or plate. Okay, this gap between the hepatocytes, this gap is actually the bile canaliculi, which means like okay, the cell membrane of two hepatocytes becomes separate from each other and becomes wide to form the bile canaliculi. This cell membrane becomes wide from one another, this thing wide from another, in order to form the bile canaliculi, and they are not lined by special cells, they are just gaps or grooves in between two hepatocytes. It is one thing that we need to remember about the bile canaliculi. Okay, so the bile is drained into the groove. Okay, they are actually even the bile, the bile that is actually produced from the hepatocyte, they will eventually be drained into the groove, and this groove will form a tunnel which is going in between many cells. So membrane of bile canaliculi also has a transporter. So around this bile canaliculi, we also have a lot of um, transporter. I'll try to make one video on the physiology of the liver, where I'll be talking about all the transporters and stuff. And has transporter and proteins, but they are different from that facing the space of DC. So here also we have one type of transporter, a few types of transporter. Here also we have a few types of transporter, but they are actually different from each other. So okay, this is actually a question of the exam. So what are the components of the bile? So there are mainly 5 components of the bile. Okay, bilirubin. The first one is bilirubin, the second one is bile salt, third one is IgA, fourth one is cholesterol, then the fifth one is phospholipid and bacchitin. So I forgot to mention here when I was talking about the space of DC. The space of DC also has plasma cells which produce IgA. That's why I said the components of the bile, one of the components of the bile is also IgA. So the space of DC also has plasma cells which produce IgA, taken up by the hepatocytes, pumped into the biliary canaliculi. Okay, from here they will actually produce IgA, go into the hepatocytes, eventually go into the bile canaliculi, and through the bile, the IgA will come into the GIT and protects the GIT from virus as well as the bacteria. Okay. Okay, the next thing that I want to talk about is the space of DC itself. So the membrane of the cell which is facing the space of DC. So okay, this part, they are actually having a different transport system as well as secretory system. For example, the breakdown of the RBC in the spleen produces bilirubin, which is transported from the portal vein sinusoid space of DC. And okay, moving on. The surface of the hepatocyte also has a transporter which can take up the bilirubin to the cell. Okay, they actually can take up the bilirubin to the cell. So after bilirubin is taken in, it is conjugated to glucuronic acid. All these things we will be talking about the physiology of the liver probably in the next videos. So after bilirubin, they will be conjugated into the glucuronic acid, and conjugated bilirubin is then actively secreted into the bile canaliculi. That is for the that is how the direction of the breakdown of not direction of the breakdown of RBC. It's actually it's like what is going to happen to the breakdown of RBC. Okay, that is for one physiology of it. The next one, drugs are also taken in by transporter on hepatocyte membrane, and then they are modified. Like for example, metabolites are actively secreted into the canaliculi, which go to GIT and combine with the fecal matter, whereas some drugs are conjugated and thrown back into blood and lost in the urine. Okay, so these are the things that you need to remember. Okay, so I've talked about the first one. I've talked about the space of DC. The second one I've talked about the multiple velar surfaces. The third one I've talked about bile canaliculi that is actually formed between the gaps. How they are formed, there's actually gaps between the hepatocytes. The fourth one I've talked about how the bile is actually drained into the bile canaliculi and what are the components of the bile. And additionally, I've also talked about space of DC. They have plasma cells which actually produce IgA. And the fifth one, I've talked about the membrane of the cell which is facing the space of DC. And I've talked about 2 examples of physiology of hepatocytes there, where what happens to the breakdown of RBC and what happens to the drugs through the membrane. Membrane that is actually facing the space of DC and all these things. They have what are the roles of these transporters for the breakdown of RBC as well as drugs. Next, the 6th one, I'm going to talk about hepatocytes. What are their function as in production of substances? Okay, so hepatocytes also have a lot of mitochondria, lysosomes, peroxisomes. Okay, and all these things are very vital in order to destroy all the bacteria that are coming from the GIT. They also have lots of RER as well as SER, that is rough endoplasmic reticulum as well as smooth endoplasmic reticulum. So smooth endoplasmic reticulum, as you know, it is produced to produce lipid as well as catalyze estrogen, testosterone, and progesterone, and it is also for detoxification too. And rough endoplasmic reticulum, it is to synthesize plasma protein like albumin, by taking the amino acid change to plasma protein, then throw back them to the blood and stuff. And we also have some Golgi apparatus there in order to modify the protein that has been produced by the ribosomes as well as rough ER. So also they also involved in the synthesis of the lipoproteins such as VLDL as well as LDL. LDS is the very bad one. They are also playing an important role in producing HDL too. So okay, VLDL that has been secreted into the blood, they act as a carrier of the triglyceride. Okay, and LDL is the main carrier of the cholesterol. So hepatocytes, they synthesize cholesterol back into the LDL and eventually will go to the blood. Okay, so that is the sixth point that I've talked about. The seventh point that I want to talk about is okay, this cell here, the stellate cells, also known as the Ito cell. So this cell usually act as fat storing cell or vitamin A storing cell, since vitamin A is fat soluble anyway. That is when they are actually being a good guy. But then this Ito cell or stellate cell actually can turn into a bad guy when there is an ongoing inflammation. So from pathological aspect, when liver is having a chronic inflammation, inflammatory mediators will stimulate these stellate cells, and they will lose their fat and becomes very aggressive and dangerous. And these E2 cells will be converted to myofibroblasts, which will start to secrete a lot of collagen, and this collagen will then constrict. Okay, when they actually form a lot of collagen in this bit of DC, they will eventually constrict the sinusoid. Okay, they eventually constrict the sinusoid. So the liver blood flow will be altered, and some areas of the liver will have less blood flow, and some areas will have more blood flow. Okay, so the part which is having less blood flow will undergo necrosis, and the blood which is the part which is having more blood flow will have this hyperplastic nodules, and that whole condition to have an alteration between necrosis as well as hyperplastic nodules is also known as a cirrhosis, where the liver becomes fibrotic and nodular. Let's say there is like more sinusoids, fibroblasts here. They will actually constrict this area. This area is not constricted, so this area more blood flow, less blood flow at this area, and this area will become necrotic. This area becomes hyperplastic nodule. So another important function of the eighth one, another important function of hepatocyte is the modification of the vitamin D. Okay, so vitamin D comes to hepatocytes in the form of cholecalciferol, and in hepatocytes, 25 hydroxylase enzyme which will do hydroxylation will form 25 hydroxycholecalciferol, and they will eventually goes to the kidney for more hydroxylation, and this will form a 125 dihydroxycholecalciferol, and this is the active form of the thing, and they can actually deal with they will cause more absorption of the calcium as well as the phosphorus. So liver disease will actually affect vitamin D metabolism too. So in the next video, hopefully the final video, I'll be talking about the second way to explain this, the second model as well as the third model of explaining this hepatocytes, that is portal labule as well as a hepatic acinus. Thank you guys.